Hello everyone, my name is Yang Chen. And so we're just gonna wait for a couple few minutes to let people come in. So hello everyone, my name is Yang Chen. I'm the organizing committee of Early Career Immunology Digital Seminar 2023 to 2024. So today's talk, we will have Dr. Wei-Le Wang from Institute of Molecular Biology and Academia Sinica in Taiwan. So the goal of Early Career Immunology seminar series, or ECI, is to increase the visibility of junior faculty that was passionate about immunologies. And one of our goals this year is to expand our reach to the people and the audience, also the speaker in Asia Pacific, including Australia and New Zealand, Japan, Korea, and across the entire regions. So this year we will have 20 talk from 2023 to 2024, and half of them will be the speaker from Asia Pacific and across Europe. And also so the talk will be every other Tuesdays and will be streamed online, and we also have a recorded seminar that will be put into the YouTube. So this year we have 9 organizing committee. 6 of them are from the US, and 3 of them, including myself, are from Asia Pacific area, including Taiwan, Japan, and Hong Kong, and Korea. Sorry. And so just a reminder, so after the talk, you can put or during the talk, you can put your question in the Q&A sections. And after the talk, we'll read out your questions, and the speaker will answer the questions online. And today's talk will be Dr. Wei-Le Wang, and he will talk about the durable lymphopoiesis and the B cells in neurodegenerative disease. And so without further ado, I will leave the stage to Dr. Wei-Le Wang, and I'll start sharing my screen.
Okay. Hello, everyone. Can you hear me?
Yes.
Okay. So first, I want to express my gratitude to the ECI Seminar Committee for inviting me to share our findings. So my name is Wei-Le Wang, and I established my lab since last October, and at Institute of Molecular Biology, Academia Sinica, Taiwan. And we are interested in topics related to immune regulation in tissue barriers, particularly the B cell in meninges and the eosinophil in the small intestine. And today, I'm going to talk about how we identify meninges, also named as the CNS borders, as novel niche for the B cell development and its implication in neurodegenerative diseases. So first of all, I just want to use this oversimplified figures to go over the life of the B cell. So we all know that B cell arise in the bone marrow. They are derived from the common lymphoid progenitor. And after a series of maturation steps, these B cells start to express rearranged B cell receptor. And when they mature, they egress from bone marrow and move to spleen. And when this naive B cell encounter antigen, they enter the germinal center for two critical purposes. The first one we call the somatic hypermutation, which allow the B cell to increase the affinity of the antibody. And the second step we call the class switch, which allow B cell to increase the diversity of the immunoglobulin. And then they further differentiate into the plasma cell. Here, I just want to highlight two critical genes. The first one is RAG1, which is required for the rearrangement of the B cell receptor. Without the RAG1 expression, the B cell development will be arrested at early stage. And the second is CD19, which is considered as the pan B cell marker to label the B cell population. So this story was coming from an interesting but unexpected observation. So back in 2016, when a group of people from Ido Amit's lab, they were trying looking for unique signature of the disease-associated microglia, they performed a single nuclear RNA-seq analysis. And when we look at this data, we notice that there's a heterogeneous B cell population, as shown here. And 3 years later, another independent group from Burkhard-Beck's lab using the CyTOF to analyze the cell in the CNS again. So according to their analysis based on the CD45 and the MHC class 2 expression, again, they were able to identify mature and immature B cell. So if you still remember what I just told you, that based on the textbook, bone marrow is the primary organ for B cell development, then you might wonder why there's a heterogeneous B cell population here. And this observation prompts us to initiate wild and bold hypothesis. Perhaps there's a niche for the B cell development in the central nervous system. So to address this hypothesis, we decide to generate a B cell reporter mice by breeding the CD19 Cree with the ROSA26 TD tomato. And then we take the brain from this reporter mice and perform the clonal section and look at different parenchymal regions as indicated as a purple color. And here you can find the imaging of the corresponding region, including cerebral cortex, choroid plexus, and dentate gyrus, and we do not find any B cell in this region. However, we're able to identify several B cells in the extravascular region of the leptomeninges. So what is the leptomeninges? So here is the anatomical structure of the meninges. Meninges are 3 protective membrane layers that surround central nervous system, including brain and the spinal cord. So as you can see here, meninges are composed of the pia matter, arachnoid matter, and dura matter. In between arachnoid and pia matter, there is an interval space we call subarachnoid space, which is filled with the cerebral spinal fluid. Meninges are also known for harboring a variety of the immune population, including dendritic cell, monocyte, macrophage, B cell, T cell, and neutrophil. So they constantly patrol this region to support the immune surveillance. However, this anatomical structure recently has been slightly revised based on the finding published on Science recently. So they find that there's an additional layer, the 4th meningeal layer, that separate the subarachnoid space into the outer and the inner compartment. And by analysis, the PROX1 EGFP reporter mice, they were able to label this subarachnoid lymphatic-like membrane, or SLIM. They also tried to inject the microsphere, which are labeled by a different fluorophore label, including the red color and blue color. And they found it can nicely separate the compartment of the subarachnoid space. This also found that the myeloid population is enriched in this slim layer. So taken together, these findings suggesting that the fourth additional layer of meninges perhaps contribute to the drainage of the lymph and perhaps also involved in the pathogen to defend the pathogen infection. But what is exactly the biological function of this additional layer remains to be further investigated. So let's move back to our story. So based on the single nuclear RNA-seq Site12 and our imaging, we want to know what exactly the composition of the B cell in meninges. So to address this question, so we decided to use a well-established approach called hard diffraction to study the B cell development. This approach was developed by Richard Hardy in 1980s. So basically, it just combined 6 different markers, including B220, CD43, BP1, CD24, to define fraction A to fraction C, and then additional marker, including IgD, IgN, to define fraction D to F. And as you can see from fraction A to fraction F, it has the corresponding to the different developmental stage of the B cell. Okay. However, when we decide to use this approach to analyze this meningeal B cell, we realized that due to the paucity of the cell in the meninges, it will be a challenge to use all 6 markers to look at each fraction. So we decided to use simplified version of the hard diffraction analysis by just using the B220 and the CD43. Then we are able to define early and late and mature B cell through the B cell development. And then we decided to look at the composition of B cell in 5 different organs, including bone marrow, brain, dura, blood, and spleen. As expected, we are able to identify 3 distinct B cell populations along the B cell development and maturation in bone marrow. Interestingly, we observe a very similar composition of B cell in brain and dura as well. In contrast, the B cell derived from blood and spleen are relatively homogeneous. We also decided to use 2 additional markers to define the characteristic of these 3 populations. So CD93 is an indicator for developing B cell. And as you can see, the expression of CD93 gradually decreases when B cell mature. And again, the expression pattern of CD93 is very similar among the B cell derived from dura, brain, and bone marrow, whereas you can find barely B cell that express CD93. And IgM is a marker or indicator for B cell maturation. Then again, you can see very distinct expression pattern for B cell derived from bone marrow, brain, dura, compared to the cell derived from blood and spleen. Then our next question is, based on the analysis of meningeal B cell in wild type, it looks like it's very similar to what we have known in bone marrow. But what if we disturb the B cell development, and what happened in those B cell developmental defect in the brain or in the dura? So to test this hypothesis, we decide to analyze again the same 5 different organs in the RAG1 knockout mice. So RAG1 is a gene known for rearrangement of B cell receptor, and RAG1 deficiency will lead to the developmental arrest at very early stage for B cell. So as expected, we do not find any mature B cell in blood and spleen. However, very similar to what we have found in the bone marrow, we can find the early B cell development arrest in the brain and dura. And here just the summary of our analysis. So taken together, we believe that meninges harbor a heterogeneous B cell population that include the different developmental stages. And seeing is believing, in addition to the flow cytometry analysis, we wonder if we can find see a real heterogeneous B cell population. So we decided to analyze the meninges derived from the RAG1 GFP mice. And here we use the light one to label the lymphatic endothelium cell. And as you can see, you can find 3 distinct B cell populations, which are RAG1 GFP single positive And as B cells start to mature, they start to express IgN. Then you will be able to find the yellow cells, which express IgN and RAG1 simultaneously. And then when B cells further mature, they will shut down the expression of RAG1 GFP signal. Then you will be able to find the single red color positive B cell. And again, it shows that the heterogeneous B cell population visually in the meninges. So we also want to understand if any behavioral difference between the meningeal B cell or the B cell that stay in the blood vessel. So to do so, we inject the tomato lectin to label the vascular structure, and we use 2-photon imaging through the skull thinning, and we are able to observe the B cell in meninges. And we also perform the time course analysis of the B cell in this region. And here I just want to show you the video. Okay, so as you can see, this B cell slow crowding in the extravascular region. You can easily identify several B cells that flow through the bloodstream quickly along the blood vessel, suggesting that there's a distinct behavior of the B cell stay in the blood vessel and meninges. Okay, so our next question is then what is the destination of this B cell in meninges? So based on the previous study, we noticed that the meningeal lymphatic vessel will be able to drain the cerebrospinal fluid or the molecular cell in this fluid to the cervical lymph node, the deep cervical lymph node. So then we wonder if the meningeal B cell can move along this meningeal lymphatic vessel to the cervical lymph node. So to test this hypothesis, we adaptive transferred 2.5 million cells from the B cell reporter mice. And then we deliver this cell through the intracellular magna injection. And 24 hours later, then we check the cell in the inguinal lymph node or cervical lymph node. And I believe you can clearly see that the majority of this cell are found in the cervical lymph node. And also we look at the imaging of the meninge of this recipient mice, then we are able to found the adaptive transferred B cell in this recipient mice, as well as the cells in the cervical lymph node of this recipient mice. So we believe that the lymphatic vessel may serve as a migratory route for B cells exist meninge and move to the periphery. So to gain more insight of this meningeal B cell at molecular level, we decide to perform the single-cell RNA-seq analysis of the cell derived from dura, blood, bone marrow without salting. And first, we just want to look at the mature B cell marker, including MS4A1, which encodes CD20, and H2AA that encodes the MSC molecule. And you can immediately notice that majority of the blood B cell express mature B cell marker, whereas only part of the B cell from bone marrow and dura express these markers. In contrast, you can barely find any single B cell that express this developing B cell marker derived from the blood. And for the B cell in bone marrow and dura, you can find a distinct region that different from this region that express developing B cell marker, including CD93, RAG1, and IL-7 receptor. So again, confirm that there's a heterogeneous population of mature and immature B cell in the dura. Then we decide to perform more detailed analysis to look at this B cell from blood, bone marrow, and dura. The most intuitive analysis just split by this B cell by tissue. So I believe that you can immediately realize that the bone marrow and the dura B cell occupy the similar territory, whereas blood B cell represent distinct region. So when we analyze this B cell and split by cluster based on the resolution that we applied, we are able to identify 10 clusters from cluster 0 to cluster 9. And you can also easily realize that cluster 0 is mainly contributed by blood B cell. Then what is the cluster 0? So here is the heat map showing you that the most enriched gene in each cluster. And based on these enriched genes, you are able to infer each cluster to the corresponding developmental status of the B cell. For example, cluster 0 is enriched with the gene related to the mature B cell marker. So we are able to infer this cluster as fraction F. And then when you look at the cluster A and cluster 9, you can easily find that these clusters are mainly contributed by bone marrow and the dura B cell. And they belong to mitotic or fraction BC B cell, which means that they are under the development. So based on this analysis, it seems like the transcriptomic pattern of meningeal B cell is very similar to what we found in the bone marrow. So now we have the flow cytometer analysis. We also have the single cell RNA-seq analysis. I guess now you believe that there's a heterogeneous B cell population stay in meninge. But what are these B cells coming from? What is the source of this meningeal B cell? Is our next question. And here is the hint. But based on several studies published several years ago, they found that the myeloid population in the skull bone marrow, they are able to reach to the dura through this channels-like structure. Therefore, we wonder if we can have the similar observation to those lymphoid populations. So to address this question, we performed a non-canonical mixed bone marrow chimera. So we took CD45.2 as the host, and we used the lead shell to cover the body of this recipient, and then we exposed the head of this recipient mice to the gamma irradiator, then adaptive transfer 2 million bone marrow cells from the donors. And we allow 4 weeks to reconstitution. So if dura or meningeal B cell are derived from the skull, then after the reconstitution, you will expect that donor-derived B cell will enrich in the skull bone marrow and dura. If not, then you will see the enrichment of the host-derived cell in these 2 regions. So here is our analysis. So the first 2 panel, we are trying to look at B and the T cell in the tibial bone marrow, blood, skull bone marrow, and dura. And you can easily find that there's an enrichment of the donor-derived B cell in the skull bone marrow and dura. And you do not find this effect in T cell. Additionally, we also look at the image, the meninge of the B cell reporter mice. By using confocal imaging, you can easily identify that the TD tomato positive cell, but IgM negative cell present in this channel-like structure. So again, taken together, we believe that the skull bone marrow provide the progenitor of the B cell that move to the meninge and finish the maturation steps. In parallel, we also perform 2 additional experiments to prove this idea. So first, we perform the parabiosis experiment by connecting the circulation of 1 wild type and the B cell reporter mice. Then we analyze the wild type based on the TD tomato expression. We know this is the parabiome derived. So normally, if you wait long enough, in theory, the chimerism will reach to approximately 50%, which means that half the amount of the B cell in the circulation will move from the B cell reporter mice to the wild type recipient. So here is what we found, that the parabiome derived B cell frequency is comparable between blood and spleen. However, the frequency of parabiome derived B cell is very limited in the brain and dura. Another experiment is that we adaptive transfer the splenocyte derived from the B cell reporter mice through the IV intravenous injection, and we wait for 1 week and to look at donor-derived B cell in this recipient. And again, you can find very comparable frequency of donor-derived B cell in the blood and spleen in general as periphery, but a very low percentage of the donor-derived B cell exists in the brain and dura. So taken together, we believe that while the skull bone marrow contribute to the B cell in meninges, there is a very limited contribution of peripheral B cell to meninges. So the next question is, okay, now we know that B cells are derived from skull bone marrow and they will finish the maturation step. But what are the key factors that support B cell to stay in meninges and finish the maturation steps? So here is the dot plot, show you that the features genes in each B cell subset and the fibroblast-like cells. And I just want you to pay attention to the CXCR4 that expressed by mitotic B cell or progenitor and precursor B cell, whereas CXCL12, which is the ligand of the CXCR4, are enriched in the fibroblast-like cell. And then we perform the niche net analysis. So this analysis basically is the integration of our gene expression profile and the non-database of the gene regulatory network to predict the potential interaction of the receptor and ligand expressing cells. And based on this prediction, I believe you can easily identify there's an interaction between the CXCL12 and the CXCR4 expressed by B cell. So to further validate this prediction, we analyze the fresh dura X-Men derived from the CXCL12 DS-rat reporter mice. And again, we use tomato lectin to label the vascular structure. So you can easily find a lot of cells that express CXCL12 in the fresh dura X-Men. And we also use flow cytometry analysis in combined with CD31, and the protolenin to define the dura fibroblast. And by getting on this dura fibroblast, we confirm that around 20% of fibroblasts do express the CXCL12 DS-rat signal. In parallel, we also look at the CXCR4 expression in early and late mature B cell derived from bone marrow and dura. And interestingly, we notice that the expression of CXCR4 is even higher in the early B cell from dura compared to the same population in bone marrow. So we believe that the expression of CXCL12 by fibroblast supports the interaction between fibroblast and the CXCR4 expressing B cell and allow this B cell to stay in meninges and finish the maturation steps. So here, I just want to use this graphic to summarize what I have just shown. So the progenitor stem cell or precursor in the skull bone marrow, they are able to move to the meninges through this channel-like structure. As they reach to these regions, they are able to interact with CXCL12 expressing fibroblast. In the meantime, this B cell expresses CXCR4. Due to the interest of time, I'm not able to tell you what we have found in aged mice. But based on our BCR clonal analysis, we know that at steady state in young mice, there's a very limited contribution of peripheral B cell to the meninges. However, this peripheral B cell disseminate to the meninges when mice getting older and older. So it sort of explains why some of the autoimmune disease prone to develop in the aged individual, because when this peripheral B cell disseminate to the meninges, they will consider those CNS-specific antigen as foreign antigen, and then further differentiate to the autoreactive B cell and produce the autoantibody. And this is how what we linked the abnormal B cell development in meninges to the neurodegenerative disease. Here, I just want to use this table to give you several examples of CNS disorder, especially mediated by dysregulated immunity, including multiple sclerosis, neuromyelitis optica spectrum disorder, and anti-MNDAR encephalitis. So these diseases are all linked to autoreactive B cell or antibody. Given that the EAE, experimental autoimmune encephalomyelitis, is a well-established mouse model to study the CNS disorder, especially mediated by dysregulated immunity. So we're also wondering what happened to this IgH MOG transgenic mice. So MOG is myelin oligodendrocyte glycoprotein. Previous study have generated this IgH MOG transgenic mice by replacing the JH locus with the rearranged heavy chain of anti-MOG antibody, such that all of the B cell of this transgenic mouse will express anti-MOG B cell receptor. And it is reported that around 15 to 30% of peripheral B cell, including bone marrow B cell, they are MOG binding B cell as shown here. So we want to know what happened of this MOG binding B cell in the meninges of this IgH MOG transgenic mice. And interestingly, we found that the frequency of MOG binding B cell reduced. And we don't know whether it's just because of the net dyslection, clonal deletion, or other mechanism that contribute to the reduction of MOG binding B cell frequency in the meninges. Nevertheless, based on our CyTOF analysis, we also observed that the frequency of lambda meningeal B cell increased in dura compared to bone marrow, although this is a poor analysis of 3 mice in each tissue or organs. But it's worthy to further investigate if the lambda B cell frequency is really elevated in dura compared to bone marrow. Also, it is important that during the B cell development, there's a critical step called net dyselection to delete the autoreactive B cell. And by using the Ig and IgD expression, then we are able to reflect the trajectory of the B cell maturation as shown here. And there's an interesting observation showing that the immature B cell during the B cell maturation accumulate in dura compared to other tissue, including tibial bone marrow, skull bone marrow. So we would like to know what exactly the underlying mechanism that drive the accumulation of the B cell maturation, particularly in dura, but not in other tissue. So in combined with the interesting finding of the increased lambda B cell frequency in meninges and the reduced the MOG binding B cell frequency in the IgH MOG transgenic mice, we would like to further investigate the underlying mechanism that control the B cell development in meninges. Okay, so lastly, I would like to thank my previous collaborator in the Kelowna lab, including Simone Brzozowski and Vincent Peng, and also other collaborators in the WashU campus. And also, I want to particularly thank the IMV core at Academia Sinica, including bioinformatics core and animal core, to help me to expand the mouse colonies. So lastly, I just want to take the advantage to advise that I'm recruiting a researcher, especially talent researchers at all levels, from student, research associate, and postdoc. So in addition to the project that I introduced, I shared with you, we are also interested to understand the function and role of a particular eosinophil subset that unique in the small intestine. So my presentation will stop here, and I'm more than happy to take any questions. Thank you.
Thank you, Wei-Le. That was a wonderful talk. Do we have any questions? I haven't seen any questions in the Q&A box. But maybe I can start the question first. I was wondering if you can I was wondering what kind of T cell are required. What's their role in the meningeal, particular CD4 T cell? Would that could possibly cause the accumulation of immature B cell, particularly in age mouse?
I if you ask me to speculate the role of T cell, which if whether they will contribute to the accumulation of B cell, I will say it will be probably marginal because based on the analysis, the number of the T cell is relative low, or at least at steady state, not only in the young mice, but also aged mice. But if you reconsider those like Alzheimer's disease model, or people have reported that the T cell, they can find T cell in the brain parenchyma. But what exactly the driver that lead this T cell accumulate in the brain parenchyma, I don't think it's clear identified. And it's not a direct effect of the T cell that leads to the accumulation of B cell. Probably there's some even in the upstream somehow that recruit the T cell to the brain parenchyma, and then T cell make something to involve the neurodegenerative disease. And also, what exactly the role of meningeal B cell in the neurodegenerative disease is still controversial. I remember recently one paper published on Nature Communication, another paper is still on bioRxiv. They have a totally opposite conclusion regarding how B cell contribute to the Alzheimer's disease development.
Mm-hmm.
Okay.
Yeah. So, I also remember there was Alexander Sadie published a paper showing that you can find those antigen specific T cell in Parkinson's disease. So I wonder if that would be a similar case. Is that possible also find those immature B cell in other brain disorder disease instead of in Alzheimer?
Yeah, it's very possible. So you have to consider what are the possible factors that either expand the B cell population or diminish the B cell population, right? So consider the homeostasis. So it requires what I just shown the CXCR4 and CXCR12 or the IL-7. So as long as in the context of this pathogenic condition, like Parkinson's disease, somehow they will be able to generate, say, IL-7 or CXL12 or BAFF, which is critical for the B cell survival. It's really possible that this factor can alter the B cell maturation or survival in meninges, even in the brain parenchyma.
Yeah, that makes sense. Do we have any questions from the audience? You can put just a reminder, you can put your question in the Q&A box and we will read it out and then we will answer it.
Well, maybe you can use a raise hand function, then probably we can unmute, right?
Yeah, that would work too. Oh, I have another question. Do you think those immature B cell will also contribute in the pediatric tumor that may not necessarily to do anything to do with neurodegeneration disease, but just in the context
absolutely. It's really possible. So if B cell development in meninges, there's some error occurred and also contribute to the B cell malignancy. It's pretty possible. So I can show you one of my experience. So during my PhD study, I studied a gene that is enriched and predominantly expressed by B cell. So when we knock out and breed with the EU-MIG, so this transgenic mice developed the B cell malignancy pretty fast. around 6 weeks, 6 weeks old, you will be able to find abnormality. And one thing which is interesting and we have no idea is that some of these mice, somehow their head were tilted. Just like it looks like they lost their balance. So I'm not sure if the B cell development is disturbed, then that will also alter the B cell composition in meninges and perhaps lead to some neurological disorders. Because the gene that we study in B cell, when we perform the single cell RNA-seq in the B cell derived from meninges, we also identify that genes again. So I was thinking, what a coincidence that I studied this gene when I was a PhD student, and I see this gene again when I was a postdoc. So definitely, if a gene that contributes to the abnormal B cell development, or occasionally the B cell development is altered or abnormal, then that might also contribute to the B cell malignancy and also in the CNS region.
Okay. Okay, so if we don't have any more questions, or do we? We don't? Okay, then I'm just gonna share my slides for the so the next talk will be on November 28th, and we'll switch back to speaker in the US and America on the 4th Tuesday of November, and at 12 p.m. EST time. And you can also find all the schedule of each talk and register online in earlycareerimmunologist.com. So please follow us on Twitter, and also if you register for each of the talk. And thank you again, Dr. Wei-Le Wang, to sharing your work with us. And thanks everybody to attend this seminar. Thank you.
No, thank you for invitation.